Hi, my name is Brian Hurt. I'm a CU sub-I here, fourth year student. And I want to talk to you today about network theory as applied to complex brain networks, neurosurgery. And, I'm going to start off, I'd like to give you guys a little background. These logos are the places I have done work at in the past, various mathematical endeavors, as it relates to many different fields, as you can see. So this is the motivation and where I've gotten a lot of my training to do this kind of research. And so I'm going to start off by a quote that's not necessarily a surprise. It's many brain dysfunctions are circuit dysfunctions. That was said by Dr. Insel, the director of the National Institute of Mental Health. He said this in a TED Talk fairly recently when talking about connectomics. So let's start with a talk with my outline here. And first I'm going to discuss what functional brain networks are actually. Then I want to discuss graph theory and how they interface in neuroscience, and specifically the evolution of these networks. And finally, touch with some neurosurgical applications. Those are the three areas I'll lightly touch on. So first of all, what is a functional brain network? A functional brain network is an effective connectivity of the different regions of the brain based on the temporal or time correlations of fMRI signals or any sort of time series signal that you get. And I point this out because contrast this with a anatomic network, which shows, think of like tractography, which shows explicit connections between nervous tissue, and this is a little different. This is more of with fMRI, for example, one voxel or three-dimensional pixel contains about 100,000 neurons, and so when you look at it this way, you're not really measuring. You're measuring sort of the aggregate activity of those 100,000 neurons, and so in this light, functional networks really they represent collections of neurons at particular locations in space and time. So tools to measure brain networks. First of all, we need the structural MRI. You guys know, very familiar with this. Next, we can get some anatomical correlates with diffusion and tractography. A lot of the mainstay of research now is functional MRI with these functional networks. But you can also get functional networks with EEG, MEG, and PET. So how do we actually measure functional brain networks? This slide shows the general process. It is fairly cumbersome and statistically and mathematically involved. So first of all, you have a structural MRI, and you've got to parcellate or essentially assign each point in that MRI to an anatomical location. And then with the fMRI or EEG, you have time series data of each of those points. And from there you can correlate those combinations of all the different points in space. And from that you can construct what's called a connectivity matrix, which is essentially a way of looking at all of the correlations between each pair of brain regions in your fMRI. So in this particular one in the upper left here, the red represents high connectivity or high correlations, and blue represents low correlations. And then further analyzing this, we can now apply anatomical relationships and some more statistical techniques to really group more of the structures together. And so in this particular slide, we have 5 different regions of the brain that have been mapped out. And from those, you can construct a functional network. And that's really what the functional network is. So now that we have a functional network, I'm going to introduce graph theory. which is the study of graphs, which essentially are mathematical structures that describe how objects interact with each other. So, and this was initially described in the 1700s by Euler when he was analyzing city planning. And so this map on the upper right here shows 4 different areas of the city with the capital letters and bridges between each of those parts of the city. You can think of those one-way bridges. And the abstract representation of that is the graph on the right with the blue nodes. And that essentially that's what the graph looks like. Now, graph theory started to really pick up steam in network theory when MySpace came out, and the engineers at MySpace wanted to analyze how people interacted with one another. So social networks became a big source of graph theory and network theory, but really it's a fairly ubiquitous concept. You can apply this to a lot of different things. So it's been applied to the internet, even as we go through, when we went through medical school, when you look at various synthesis pathways, that can be viewed as a graph, in graph theory. I know we view those as feedback and back propagation and all this stuff, but this can really be viewed as in the light of graph theory. Just the basic properties of graph theory. First is structure, and this upper right schematic shows a continuum of types of structure. So you have a regular structure, which I think is the easiest to understand, and that's essentially each node or neuron in that network is connected to more or less the same 4 or 5, or really a finite small number of local nodes. And so if you think of it in terms of information transfer, to go from, say, the top of this network to the bottom of the network, you have to go through many intermediaries to get to the bottom. Contrast this with a random network where you have all your nodes are randomly connected to other nodes, and it's irrespective of the length. And so while information can travel from one end to each end, if you will, of the network fairly easily, it doesn't come without a cost. If you think of these as sort of roads, the cost of building all these roads is fairly expensive, if you will. And then we have in the middle, we have a sort of a happy medium. We call it a small world network, and that's a mixture of both. And the concept behind a small world network is you have parts of the network that are regular and parts that are random. So you have the key thing, you have what are called hubs that can connect fairly distant portions of the network. And so with that, you can then think about, well, if you can just get information to a hub, then it can span large distances and get across the network too. And it's a fairly efficient way to do this. And I'll hammer this home a little in the next slide. And to discuss maybe some nomenclature of networks, and I've discussed hubs, and that connects 2 kind of areas, and we have 2 in this picture on the left. We have clusters of nodes that are circled here and that are connected by a hub, and within each cluster, each neuron is more or less connected just to itself. There's no connection outside between 2 modules other than that hub. And so, and now think of how information may travel. And one way to look at this is that red line. And to get from that initial neuron in the top there to the bottom, it has to go through 6 different neurons to get to reach that 7th neuron at the bottom. And so that's kind of what we talk about as path length, and that it's synonymous with efficiency. So the shorter the number of connections it takes to get to another node, the more efficient the network is. And when we apply these to patients' networks, we've it's been shown that the brain at baseline, in terms of fMRIs, is already organized along functional boundaries. And so that means, if you're doing a certain task that's measured on fMRI compared to not doing a task, or there's a baseline functional network that's a small world network. And that is the substrate for which that's kind of the current thought of how the brain transfers information. So I'm going to talk a little more about small world networks. And on the right here we have the U.S. interstate system. And that is more or less like a regular network, meaning each city in the interstate system is connected to its local cities around it. And so if you were to drive from LA to New York, then you'd have to drive to Las Vegas, Salt Lake City, Chicago, and so on and so forth. And so you go through several different hubs or cities to get there, because each city is more or less doesn't have long-range connections. Now, contrast this with the airline flight paths, if you will. And this is more of a small world network, and this is where the term hub came from, really. And this is fairly obvious. We have airport hubs, and so if you were to fly from LA to Albany, New York, you'd fly from LA to New York City to Albany, or something along those lines. But really, it would just be one connection. Imagine if you had to go 4 connections to get to where you needed to go. We would be very unhappy. Back to brain networks, though, and so now we're going to start to really apply graph theory to brain networks. In the schematic here, all I have is these are different ways to view a functional network. And so how can we use them? Graph theory, I've talked about how information flows. That is really how graph theory helps us. We can just quantitatively describe how information will flow through a network. So as an analogy, so the network dynamics, so structure and function are synonymous. So the way to think about this is, the structure of the network facilitates how information flows. I've talked about that several times over now. But really, as in many networks, as the information flows, the network changes the structure to accommodate that information. And so structure and function become innately coupled. And a river is a good analogy for this, because rivers start out as a stream. That stream starts to modify the landscape. And as more water, that landscape allows more water in as it erodes down. And then that water then in turn erodes the structure of the basin even more, and you get a larger river. Now I put a dam here because that is a way to drastically modify the network, and it modifies upstream and downstream. And back to the brain. So how do these functional networks change in the brain as a function of age? A study in 2010 was put out, and they analyzed 2 groups of people. They first analyzed 79-year-olds, and then they analyzed 21 to 31-year-olds, and they looked at the change in those functional networks. And as 79-year-olds, a lot of their networks are more or less oriented anatomically, and this graph is pseudo-anatomic in the sense that, anterior posterior is preserved and there's some right left laterality to it. And then they looked then at the 21 to 31-year-old group, and there was becoming more long range or more hub-like behaviors that would come from just development. And so these are statistically significant changes, and that's what's shown on the right, the far left here. So conclusion is functional brain networks change over time. But how? And how can we show this? So this brings me to my research. I was curious, and I wanted to investigate how small world networks can arise from just local interactions of neurons. And the way I try to begin answering this question is I thought I would build a mathematical model that used Hebbian learning as a way to analyze. So the mathematical aspect of it is fairly simple. The behavior of neurons is fairly simple. It just takes the sum of all the inputs, and if it is above a certain threshold in that neuron, then it fires a postsynaptic action potential to its neurons that it's connected to. And I went a little further, and I decided to propose an experiment, if you will, a computational experiment. I decided to create a computational mouse. And each mouse has internal neurons, they have motor neurons, they have olfactory, sensory neurons. And I've put this mouse in a computational environment where I put cheese, and that cheese emanates a smell. And these mice can move around in their environment, and if they reach that cheese, they get some energy. And if they don't get the cheese, then they die off. There's a finite amount of energy that each computational mouse can expend. And so, with that, you end up sort of developing an evolutionary simulation that selects for those computational mouse configurations that give you the best behavior. So I want to analyze those adapted mice. And so that's essentially what I have done. And here's an animation of one animation that I did. And before I play it, on the upper right we have the network itself. We have 50 internal neurons. We have 4 motor neurons and 4 sensory neurons. And on the upper left we have the environment with the cheese, and that's the field that the cheese emanates, if you will. And on the bottom, the long chart is the connectivity of neurons. It's a histogram, so it shows how many the connections between each neuron. It shows the number of connections for each neuron. And if you recall, back in the small world networks, we had a power law distribution where most of the hubs, or most of the neurons in the network, were actually not hubs. And so what you're seeing here is there's a lot of connections. Most neurons in this particular setting have 30 to 45 connections. And so if that's not a small world network. And on the bottom right, we have that connectivity matrix that I discussed early on, and that will, as the animation goes on, that reflects the Hebbian learning that's been going on. And there's both positive and negative learning, so synapses can be removed, but synapses can be strengthened as if they fire and wire together. So I'll start this animation. So the animation goes, and you can see that as time progresses on, neurons are losing their synaptic connections. Most of them actually only have 2 to 5, and then there's a handful that continue to have, on the order of 10 to 20. And that's reflected in this connectivity matrix on the lower right. Now, the mouse did not find the food, but I think the illustration still shows that we can get these small world networks if just using simple Hebbian rules. Now, there's some more work to be done, so don't take that to the bank. So I'm going to finish off with some neurosurgical applications. So in TBI, mild TBI, structural MRs don't particularly show, aren't very sensitive at showing the extent of damage. So these researchers in 2014 decided to look at fMRIs and construct functional networks. And what they found was they did find some sensitivity. So they found that initially, after the TBI, the subset of patients with a TBI had increased average path length or reduced efficiency. And then the cost of it, which is another measure of efficiency, was also decreased. And they chose to use modularity as one of the surrogates for this. The second one is DBS effects in Parkinson's. The researchers in 2014 wanted to look at how functional networks change before and after DBS implantation. So this particular patient had right subthalamic implant, and they evaluated his fMRI before and after surgery, and they found their conclusion that it was a couple. And they found that DBS promotes a more stable global network, and that's evidenced by the graph on the upper left. And you said the red line compared to the black line. This essentially represents it's more stable. It's not stable as the controls are, but it's in the right direction. And the second thing they did was they looked at the efficiency of the nodes. They can do the nodal efficiency, if you will. And these blue dots represent an increase in over 20% or more of nodal efficiency in the right hemisphere. And this suggests that the brain networks are reconstructing to more of a normal configuration. And the last one is more of a perspective discussion. It's about intraoperative planning in neuro-oncology. And so this graph theory, it's just a new language we can use to further understanding of how the brain is functionally organized. And it's also been studied that functional networks in patients with various brain tumors are grossly altered. And so the problem, I think the barrier to using this, though, and intraoperatively is, it's a necessity to relate these disruptions to some sort of symptomatology or function of the patient, and that still remains to be seen. But in the end, though, one of the strengths of this graph theory is we can model the effects of taking a certain region out of the brain and trying to analyze how the brain will compensate for this, and then ultimately try to predict the effects of a surgery on the network. And so I'm going to end there. These are my two mentors, Dr. Ojiman, Dr. Newton. Any questions?